So it's my pleasure to welcome you to the 2019 Daniel I.C. Wang Lecture on the Frontiers of Biotechnology. And I wanted to first say a few words about the named lecturer. The person that this lecture is named after is Professor Daniel I.C. Wang, who is here with us today. We're very happy to see. I'm going to give you a little bit of history about Professor Wang. He received his bachelor's degree, his SB, in 1959 and his master's in 1961 from MIT. And his PhD in chemical engineering from UPenn in 1963. He then spent 2 years in the US Army at the US Army Biological Labs at Fort Detrick, Maryland from 1963 to 65, and he joined the MIT faculty in 1965. And I'll pause here just to give you a picture of the young Dr. Wang, and also in his military gear. Professor Wang founded the Biotechnology Process Engineering Center through the NSF Engineering Research Center Initiative and acted as its director from 1985 to 1998. He also ran a summer short course, which was the longest running and most successful. He was subsequently named as an institute professor of chemical engineering at MIT in 1996. And now this is actually a very notable thing because we only have a few institute professors. There can only be a certain number, and they are the highest level of recognition that you can receive at MIT. So we are very fortunate to have our own institute professor, Wang. Now in 2000, he was also given the Temasek Professorship at the National University of Singapore, where he devoted part of his time to helping Singapore in their biomedical science research and development. Over his lifetime, Professor Wang has received many awards, including those from the ACS, including the Marvin Johnson Award and the David Perlman Memorial Lecturer. He's also received several awards from the AICHE, American Institute of Chemical Engineers, including the Food, Pharmaceutical, and Bioengineering Award, Institute Lecturer, and William H. Walker Award. He was given the Amgen Award in Biochemical Engineering from the Engineering Foundation, and he's also received the Asia Pacific Biochemical Engineering Award. Professor Wang has been elected to the American Academy of Arts and Sciences, the National Academy of Engineering, the American Institute of Medical and Biological Engineering, and Academia Sinica, which is the honor society of the Republic of China, located in Taiwan. He's also a member of the International Institute of Biotechnology. He's consulted for over 50 companies worldwide and is on the board of directors and scientific advisory boards of a number of public and private companies. During his lifetime, he's authored and co-authored 5 books and over 230 papers and 15 patents. Professor Wang is recognized as one of the fathers of biochemical engineering, and we're very proud to have him as our institute professor in chemical engineering. Now, this is a great opportunity to recognize the sponsor of this lecture, which was originally simply called the Frontiers of Biotechnology and was renamed the Daniel I.C. Wang Lecture on Frontiers of Biotechnology, thanks to a gift from Dr. Nubar Thien. And I'm going to tell you a little bit about Nubar, who is also one of our alum and represents the next generation after Danny of biochemical engineering, and in fact has quite a history here in Cambridge. So Dr. Nubar Thien is the sponsor of the DIC Wang Lecture and one of our own alumnus. He entered biotechnology during its emergence as an academic field in industry, completing his doctoral work in biochemical engineering here at MIT in 1987. During his career as an inventor, entrepreneur, and CEO, he has co-founded and helped build over 40 life science and technology startups as the founder and managing partner and CEO of Flagship Ventures. Nubar has essentially been involved in several important startups, and these include, he was founder and CEO of Perceptive Biosystems, a leader in bioinstrumentation that grew to $100 million in annual revenues. After Perceptive's acquisition by Perkin-Elmer, Applera, In 1998, he became Senior Vice President and Chief Business Officer of Aplera, where he initiated and oversaw the creation of Celera Genomics. He's written numerous scientific publications and is the inventor of more than 80 patents. He has been a lecturer at the Harvard Business School, and from 2000 to 2016 was a senior lecturer at MIT's Sloan School of Management. He teaches and speaks around the world on topics ranging from entrepreneurship, innovation, and economic development to biological engineering, new medicines, and renewable energy. He is a member of our MIT Corporation and a member of our department's visiting committee. So he's been an outstanding alumnus. He has also received a Technology Pioneer 2012 award from the World Economic Forum in recognition of breakthrough solar fuel technology. And he has served as chairman of the Global Agenda Council on Emerging Technologies of the World Economic Forum. Nubar is a passionate advocate of the contributions of immigrants to economic and scientific progress. Nubar received the Golden Door Award in 2017 from the International Institute of New England in honor of his outstanding contributions to American society as a U.S. citizen of foreign birth. He was also awarded a Great Immigrant Honor from the Carnegie Corporation in 2016 and received a Technology Pioneer Award from the World Economic Forum in 2012. He was presented with the Ellis Island Medal of Honor in 2008. Now I'd like to introduce our honored speaker. Dr. Michael Tien had received his bachelor's in chemical engineering from Caltech in 1982 and his SCD from MIT in biochemical engineering in 1988. For those of you who don't already know, SCD and PhD are equal. He then did a postdoc at the Whitehead Institute across the street and joined Merck Research Labs, working in vaccines and recombinant proteins. Mike was process development leader and plant startup leader for the first effective commercial HIV protease inhibitor, Crixivan, for which he was given the division's top recognition. And he made key contributions to Merck's Januvia, a treatment for type 2 diabetes, and Merck's efforts in quality by design and knowledge management. Mike has held various positions within the research and manufacturing divisions at Merck, including Senior Director of Chemical Pilot Plant Operations, Executive Director of Chemical Process Development, Vice President, Process R&D. In 2005, Mike then co-led a team to redefine the paradigm by which Merck brings new drugs to market. This effort resulted in the creation of a new function at Merck, the Global Pharmaceutical Commercialization Organization. This group included engineers and analysts from both R&D and manufacturing, and reported up through manufacturing. In 2005, Mike was appointed to head this group. He later served as Senior Vice President of Global Science, Technology, and Commercialization, and also took on responsibility for technical support of commercial sterile operations. In 2013, Mike's role added new product development and inline support for therapeutic proteins, vaccines, and animal health products. In 2017, Mike was named as head of the end-to-end operating unit for biologics, therapeutic proteins, and sterile operations. Mike currently leads the Manufacturing Systems Design and Commercialization Group, which is responsible for the introduction of new processes and methods for new vaccine and biological products, new product technology transfer, deep scientific and engineering support for inline products, new facility introduction and startup, and development of the next and best practices for new facilities. Mike has made numerous invited conference presentations and guest lectures on the pharmaceutical industry and chairs the advisory board for the Department of Chemical and Biomolecular Engineering at Tufts University, our neighbors just down the road. So with a great deal of anticipation, I am happy to introduce to you Dr. Michael Tien, and we're going to hear a little bit more about his technologies. Thank you.
So good afternoon. I'm honored to be today's speaker in this lecture series, and particularly since this series is named after Dan Wang, who is here today. And I want to make sure that I don't offend somebody. Alan Hatton's here as well. Both were my thesis advisors, so I want them to both feel good. And also, I want to thank Nubar for the sponsorship of this series. Nubar and I were in the entering class together And I have to say, Nubar, where's Nubar? He's back? There he is. Great job on the snacks today. Much better than the brownies and cookies we had when we were graduate students. So thanks. One other note, I noticed that Professor Hammond was trying to get people to sit in the front row. Some of you did. So you're not as smart as the folks back there because I spit, so be careful. This series called Frontiers of Biotechnology, I think that's a really great title because it allows a lot of leeway in terms of where people want to go. And as I think about frontiers, I think about tales, I think about stories, right? And you can imagine some stories of the biotechnology frontier are very deep, down to the atomic level. Some focus on technology platforms. There are a lot of different tales one can tell. Today's tale, told in two parts, will be focusing on complexity and on speed. And as practicing engineers in an industry where one tries to serve the needs of the patient, speed and complexity are always things that we have to address head on to ensure that the innovations that come from research don't remain laboratory curiosities, but actually get to the patients that need it most. So that's where we're going to focus today. Now, as I think about the way we do our work, it's important to think about the context in which one's work is done. Right? We don't simply do development work in isolation. We do it in the context of what's happening in the world around us. When I think about the biopharmaceutical industry and medicine, and when I think about what's happening in the world around that industry, I think what would be a good way to put us in the right context? And so I'm going to start today with a quote that even though it's about France, Paris, and London in the 1780s and 90s, written in 1859, I think you'll find it is strangely apt today, right? It was the best of times, it was the worst of times. It was the age of wisdom, it was the age of foolishness. It was the epoch of belief, it was the epoch of incredulity. It was the season of light, it was the season of darkness. It was the spring of hope, it was the winter of despair. We had everything before us. We had nothing before us. We were all going direct to heaven. We were all going direct the other way. Right? Our industry faces huge challenges and yet huge opportunities. And so, as I said, I want to tell you a couple of tales, a tale of complexity and a tale of speed that exemplifies the opportunities ahead of us as well as the challenges. Now, in order to tell a good tale, you need villains, and so we're going to have villains in today's tale. And today we have the so-called king of maladies, cancer, as our key chief villain. And I'm going to talk about cancer in two different perspectives. We all know that cancer, when we use that word, it sounds like it's a monolithic word, but we know there are many, many different types of cancer, each needing their own specific type of treatment, and I've laid out the various different types of cancer shown here in the big wheel. One of the emerging knowledges from biomedicine of the last decade is the fact that tumor cells, many tumor cells, have developed the means by which they can camouflage themselves from the immune system. Our T cells have on them something called PD-1 receptors, which are used to downregulate T cells so that T cells don't do damage within the normal healthy human body. But some tumor cells have developed the means to address those receptors, turning off these T cells, effectively acting as camouflage from the immune system. And in the last 5 to 8 years, the big revolution in oncology is how can we find ways to turn off the camouflage mechanism, allowing the immune system to attack cancerous tumors. And so we'll talk about that as our second story. But in our first part of the tale, I go to the gray section of the pie chart. And in that gray section of the pie chart, not specifically listed, are HPV, that is human papillomavirus, related cancers. Cancers of the cervix, penile cancer, oral cancer, anal cancer. A wide variety of cancers is listed in the table over in the lower left. These cancers are caused by the villain over here on the left, the human papillomavirus. This virus can cause both precancerous and cancerous and cancers as shown here. Now, when we talk about cancer not being monolithic, HPV isn't monolithic either. There are many, many different serotypes of HPV, but if we look at those serotypes, not all cause cancer, Many will cause herpes, but not cancer. But I've listed here the contribution of the various serotypes as cancer-causing serotypes of HPV. There's something that we should draw away from this. First, that the virus itself is complex, can cause a lot of complex issues. But secondly, that if we are to effectively attack HPV-related cancers, we can't attack just one serotype. We have to attack many. Now, when we think about vaccines, and we're going to talk about vaccines as our means of defeating this particular villain, we think about the spectrum of opportunities we have to provide treatment in humans. And certainly small molecule, organically based compounds have been the mainstay of the biopharmaceutical industry for years. Although vaccines have been around for, since the 1800s in an organized fashion. And we can look at the size of these various types of therapeutics and see that when we get up to the things that are most commonly used for vaccines, that they are large, they are very, very complex, and challenging to deal with, right? And some of them are not able to pass a sterilizing membrane and thus need to be produced aseptically. We're going to focus on this right here, the human papillomas virus-like particle. So this is not the actual virus, but instead is the proteinaceous capsid within the virus, which in the virus is contained. This capsid we're going to talk about a little bit more in terms of what constitutes this capsid, but I'm going to go back to the fact that this is not a monolithic disease, that there are many different serotypes, and that we have to make sure that whatever we're developing as a vaccine is effective against enough of those serotypes that we can do what we need to do against the disease. Now, when HPV vaccines first came out in the mid-2000s, there were, depending upon the vaccine, 2 or 4 serotypes contained in the vaccine, which addressed about 70% of HPV-related cancers, about 50% of the precancerous states that HPV could relate to. Over the next decade, it was determined that that was not enough, right? That if we really wanted to do our best against HPV-related cancers, we needed broader coverage against the serotypes that could cause cancer. And so Merck developed a vaccine that came out in 2014. This vaccine has 9 serotypes. It provides 90% coverage against HPV-related cancers, and that coverage is global. So it addresses all of the serotypes that can be found around the world, and it provides 80% coverage against the precancerous state. And so that is what we're going to talk about today as our means of addressing HPV, and talk about the complexity when dealing with this as a vaccine. So let's take a look and see what makes up a virus-like particle. This virus-like particle is made up, as I said, of proteins, and it turns out it's made up of just one protein. We call it the L1 protein, that self-assembles into what we call a capsomer. So that's a pentamer of the 5 L1 proteins. These capsomers, in turn, assemble into this virus-like particle. And so if you want to think of capsomers as like the panel on a soccer ball, and now you need many of those panels to be constructed in the right way to make an effective soccer ball, that's our virus-like particle. Now, as I mentioned, the most recent vaccine has 9 of these serotypes in it. And it turns out that to have 9 of them, you have to go through 9 different processes by which you make these virus-like particles. And so we'll delve a little into the challenges of that. You might ask, well, how do you go about making one of these? So this is something that the L1 protein is cloned into yeast in this case. The yeast is grown up. We make a frozen cell slurry of the yeast cells. They're then purified, and the L1 protein-based virus-like particle is then absorbed on an alum adjuvant in order to stimulate the immune system. And that then is formulated and filled. Please note that in order to make the full vaccine, you need to have all 9 serotypes present. So think about that as a manufacturing problem. Just kind of put that back in the back of your head. We'll come back to that complexity in a minute. But you have to do this piece of it 9 times, 1 for each of the different serotypes. So that's what we're talking about today. Note that for these serotypes of HPV, there's roughly between 70 and 90% homology between these different serotypes. And so that means that these spheres The average is about 72 of these L1 monomers. The L1 monomer obviously is different for each one of the serotypes, and as a consequence, these virus-like particles are slightly different for each one of the different serotypes. So let's get into development. So now we have to first do our yeast fermentation and create the virus-like particle, or at least the L1 protein. This goes through a very typical sequence, as you might imagine. So a lot of work done at the bench, cell line selection, looking at fermentation conditions, testing those conditions out and iterating on those conditions at the 20-liter scale, going to the pilot scale where one wants to make one's clinical material, finally going to a launch scale, and as one expands the market, expanding to a larger scale. Doing the fermentation development on this, you might think would be straightforward. How different could it possibly be to maximize the amount of cells, to maximize expression of the L1 protein, and to get a good quality protein? And it turns out it was quite difficult to do the fermentation optimization here. And I showed this column as number 1, the first serotype that was developed. And I should say these first 4, which came out in the original Gardasil vaccine, which was approved in 2006, these first 4, the development occurred between 1994 and 1998. Now, that's about 8 years before the vaccine was approved. And the reason there's that significant time lag is this process needs to be developed and locked before we make the phase 3 material. And for a vaccine of this nature, that phase 3 material needs to go into tens of thousands of patients worldwide. And so you can imagine these clinical trial times are long. At these patients, we need to have suitable follow-up to prove durability of the vaccine. That is, that the immune response lasts for long enough that we can feel comfortable that we've given lasting protection. So this development work happening in the early to mid-90s, again, starting with this first serotype. And I think the folks who worked on this were surprised by the amount of variation that was needed in the parameters. And I've shown the various fermentation parameters that were looked at in terms of optimization of this fermentation process. I highlight in red the glucose feed. It turns out that start time, where you started this glucose feed, how long it was, and what kind of feed profile you gave to the various serotypes was critical for not only the number of cells that you grew up, but for protein expression and the quality of that protein. And so this was very carefully looked at for each and every one of the serotypes. As I said again, we started with this first serotype, we then iterated to the second. It became fairly obvious as we went through the development of this second serotype that looking at each one of these parameters, while being helpful in creating optimal conditions, we really wanted to move more towards a platform approach. But with the pressure that was on the team to deliver the phase 3 materials so that we could get these very, very large studies going, we did our best to iterate on these last number 3 and 4 and get this material into the clinic. These conditions were what then went on to commercial manufacturing. Then in the 2000s, we began development on these 5 strains, these 5 serotypes. And again, trying a platform approach. And you can see the platform approach was much more effective for many of the parameters. But again, a significant amount of variation between the different serotypes to optimize for cells and protein expression. The teams that did this development work, both in the 90s and the 2000s, came to the same conclusions. First, that the culture media and operating conditions required really tight control. It turned out that the natural variation we saw in media components and the ability to control the process to these rigorous parameters proved really, really important to get the right amount of protein expression. We certainly tried to use lessons learned from these early serotypes to inform how we went about going development for the later serotypes. And significant process optimization, as you can see from the table, was required to get these optimal parameters of, or optimal outcomes of amount of cells and expression in the cells, as well as the quality of the protein going into the vaccine. The team, you know, when I went back and I talked to the team in particular that did the first development on the 4 strains, and you know, the one thing that they had is kind of their motto was one size does not fit all. They themselves, even when they look back on what was required to develop the different serotypes, are surprised at the amount of variation in the different serotypes that were required to get the optimum condition just to get the cell slurry paste. And now at this point, at the end of this step, we just have paste that we now have to process. We have vesicles to make, or virus-like particles to make. That then goes to the downstream. And for the downstream, the first materials made for the clinic, the phase 1 material, I have to say we weren't really taking a very sophisticated approach. This was more of, we now have these virus-like particles that we've been able to make. Will they really have activity? Will they really stimulate the immune system? Can they act as a protectant against HPV? And so speed was of the essence for getting things into phase 1. And as a consequence, the process doesn't really look like the process we finally ended up at launch. One of the things that we discovered as this was being expressed in yeast is that crude virus-like particles are actually formed on the first expression in the nucleus. And we'll take a look at these crude particles, but it was these crude particles that we actually put in to the phase 1 clinical trials. And I'm going to say that as when we take a look at them, you'll see they're really ugly looking, right? They don't really look like a lovely soccer ball. They look like maybe a soccer ball that's been in far too many soccer games. But we put that in the clinic, and when we saw initial effectiveness in the clinic with these very crude vesicles, we were excited. But one of the other things we found as we went into phase 1 is that these very crude virus-like particles had very poor stability. They fell apart relatively rapidly, and even at room temperature, we saw a significant decline in the formation or the maintenance of these virus-like particles. And so one of the things that we set about to do is we went around the production or the process development for launch and commercial production was to say, how can we make more perfect virus-like particles that will have high stability? Now, when I talked about the virus-like particle here, one thing I forgot to mention when I first described these particles, yes, it's a bunch of L1 proteins that form from the monomer to the capsomer to these particle-like structures. But one of the things that you can't see here is that between these capsomeres are disulfide bonds. There are a variety of different types of disulfide bonds. For some of these neighboring capsomeres, there's just one bond between them. Some will have 2 disulfide bonds, one going in each direction. And in other cases, we will have disulfide bonds going across many of these capsomeres. These disulfide bonds, it turns out, were critical for the stability of the virus-like particle. And so while we created crude virus-like particles with this initial process did not have these disulfide bonds, or many of these disulfide bonds, as we needed for stability. And so this launch and commercial process needed to do 2 things. Number 1, it needed to purify this L1 protein. And number 2, we needed to reassemble these virus-like particles and ensure that we got the disulfide bonding right so that we had stable virus-like particles. And you can see that the process, as we went from the initial clinical process to our launch and commercial process, that we added a variety of components. We needed to chew up more of the DNA and RNA, and so added an RNase treatment. We needed to make sure that we took out, with a low porosity microfilter, a lot of the solids that were present as part of the yeast fermentation. From our capture chromatography step, we added a multiple pass chromatography to make sure that we purified things as much as possible, of polishing chromatography, and most important, and we'll talk about this in more detail, these crude virus-like particles, there was no way that we could fix them as they were, and so we went through a disassembly process followed by a reassembly to recreate these virus-like particles and then induce the disulfide bond formation so that we could get stable virus-like particles to be able to use for commercial production. We've made it a little more sophisticated, our adjuvant absorption process. The key piece here is that while we had a lot of variation in the upstream process, one of the things from the very earliest conception was to ensure that we had a platform purification process, because we didn't want to have to do a lot of variation in the downstream processing of these virus-like particles. And so we developed common operations across this. Secondly, it was noticed early on that we got significant variation in the feed to the downstream process. That these different, you can imagine the different fermentation conditions that went on, the differences in the amount of stuff that we would get downstream. And so this downstream process had to be robust enough to be effective for all of the different fermentations, for all of the different serotypes. And so from the very beginning, that was one of the goals of creating this downstream process, was to create something that was highly robust to the differences that we would see. One of the things that we did not expect, but came out to be true, is that in fact, small variations in membrane and resin, this was lot-to-lot variation, could have significant impact on the recovery of the virus-like particle and its quality. The non-specific interactions between the virus-like particle and the membranes and resin proved to be particularly challenging, and the process had to be designed to make sure that we could withstand not only variations in the feedstock to this downstream process, but also to the natural variations we would see lot to lot in membranes and resin. The final product of this process, and remember that it is adsorbed onto adjuvant particles, in this case a monoaluminum phosphate. The final product of this process is a colloidal suspension. This colloidal suspension has the monoaluminum phosphate particles that has these virus-like particles adsorbed to it. While this was now stable for use in conventional vaccine application, it is still susceptible to extremes of temperature, handling, and storage, right? These stable virus-like particles are essential, and so we had to make sure that our conditions for handling and storage, as well as actual giving of this vaccine, adhered to certain temperature ranges to make sure that we could ensure a stable product given to our patients. So let's talk about disassembly and reassembly. This is really the most important part and the most challenging part of this process. This is what I meant by ugly, right? So that's actually what's naturally formed in the nucleus of the yeast, this native virus-like particle. As I said, its stability isn't great, and if you look here at the size exclusion chromatography chromatogram, you can see that there is a whole set of aggregates. So while this is the prettiest picture of the lot from that comes out of the nucleus, there are a whole host of aggregates of many different sizes that come out of the fermentation. Now, what we do is we take these ugly virus-like particles, and they're first subjected to a disassembly process. We know that these virus-like particles first assemble, and that assembly is aided by a higher ionic strength. And so one of the first things we do is we reduce the ionic strength. The other thing we have to do is we have to make sure that none of the disulfide bonds that may have been made are still present. And so we have to add a reducing agent, in this case DTT, to break up the disulfide bonds so that we get capsomeres and finally the L1 protein in solution. And once we have this disassembled solution, we now have to reassemble. And reassembling is not trivial. It's not like, well, let's just change the conditions, put some salt back in, get rid of the DTT, and we can all have a good day, right? What we found early on in the development of the reassembly process is that the rate at which you bring back the ionic strength, the rate at which you get rid of the antioxidizing agent is absolutely critical to getting spheres rather than tubes. So if we do this too fast, we get long tubes of the L1 protein rather than the virus-like particles that we need to get for this vaccine. And so this disassembly and reassembly process, the rates at which we remove these reagents and add them back in is absolutely critical. Now imagine doing this at the large scale. We do this via diafiltration. And so the rates through these diafiltration membranes have to be very carefully controlled as we go through the process. The buffer content has to be very carefully controlled to make sure that we are bringing on or off the ionic strength, having the reducing agent or relieving the reducing agent in a very controlled fashion in order to get that virus-like particle. And you can see from the size exclusion chromatography, we get just the particles, no aggregates in the final product. That all sounded, at least when I was listening to it, sounded too easy. And let me assure you, whether it was the fermentation development, the downstream development, the disassembly, reassembly, very, very challenging process. And perhaps the most challenging is the disulfide bond formation, right? Certainly taking away the reducing agent is important. But we know that there are other environmental factors as well that catalyze the formation of disulfide bonds, and we have to make sure that to the extent possible, we're controlling any other extraneous factors that can control disulfide bond formation. When we first did this, when we first put out our Gardasil vaccine, we didn't know what was important and what wasn't. So how many disulfide bonds do you need? Do they need to be all trimer? Do they need to be dimer or monomer? Well, what exactly do you need? We didn't know what we needed. We knew what we had and what worked, right? And so we set specifications based on what we had and what worked. The challenges we got from agency was, hey, agencies was, this is a complex quaternary structure. How do you make sure that all the parts of it are right? And so while we had biochemical engineers and biologists working on and developing the process by which these virus-like particles were made, we had to have a whole separate group of bioanalysts developing an entire toolkit by which we could interrogate the primary structure, the secondary structure, the tertiary structure, and the quaternary structure to prove to ourselves and to agencies worldwide that each and every time we're making the right virus-like particle, the particle that can relate back to the data we had from the clinic, showing that this would be an effective vaccine. And so we developed the toolkit that is shown here, looking at primary, secondary, tertiary, and quaternary structures, and even the virus-like particle as absorbed on the aluminum adjuvant. Those tests that are in bold are the tests that must be performed on every lot produced. And it was very important to agencies worldwide, in particular, that we have something that confirms the primary structure, that confirms the tertiary and quaternary structure, and that finally confirms the potency as the adjuvanted virus-like particle. All of the tests shown here that are not in bold are what we call extended characterization tests. Anytime we make a change in the process, anytime we have a process deviation, we must do the entire battery of extended characterization to show that we have not changed the virus-like particles in the process. So I hope that gives you a sense of the complexity that surrounds this vaccine, this 9-valent vaccine. I think we can kind of finish this part of the tale by talking about where we are now. And we talked about the process engineering, the molecular design, the bioanalytics. What about this is actually an effective vaccine? When we came out with the original Gardasil, as I said, it only had 4 serotypes. It only provided 70% protection against cancers, 50% protection against the late-stage precancerous state. With the advent of Gardasil 9, 90% protection, it was interesting. All of a sudden, the world kind of woke up in 2017, 2018, and said, wait a minute, what do we have here? And it was actually the Canadians first who said, this provides enough coverage that we can, and dare we say it, we can eliminate HPV-related cancers. And what was amazing was their statement that they decided to create an immunization program where they wanted everyone in Canada of the appropriate age to be immunized so they could eliminate cancer resonated throughout the world. It was only shortly thereafter that New Zealand and Australia started similar programs. And when New Zealand and Australia started their programs, WHO said, here we have the mechanism by which we can globally, for the first time, we can eliminate types of cancer. And so the call went out, and as you can imagine, that's a pretty loud call. There's only one vaccine that has the 9-valent vaccine, and that's our vaccine. And so the call went out for a lot of this vaccine. Now imagine in 2017, we made 17 million doses. By 2024, we'll need 250 million doses. So as you can imagine, the supply chain that we've currently created to make this very complex vaccine is not adequate, and we're in the process of a $2 billion investment to create and blow out our supply chain so that by 2024, we will be able to help the world eliminate HPV-related cancers. And as a side story, and I think this is fascinating, and this kind of goes to what happens in the complexities of the world. About 2 years ago, China decided to radically change its policy with regard to medicines and vaccines. Prior to 2 years ago, the Chinese bureaucracy, regulatory bureaucracy, was extremely slow. And if you looked at the average approval timing of the drugs that were being approved by the Chinese authority prior to that, from when those drugs were approved in the US and Europe, as an example, the average difference in time was about 10 years. Right? About, as I said, 2 years ago, China woke up and said, no, we really want to get the very best in medicines available. And as the premier at one point last year was walking through a hospital ward, talking to the person who was running the ward, he said, so what is all this? And the person who was giving the tour said, these are all women who are waiting to die. In China, about 100 women die a day of cervical cancer that's HPV related. And the person giving the tour related to him that it was sad because these women didn't have to. There was a vaccine. There's a vaccine? Yes, there's a vaccine. And it was interesting, 2 work days later, we got a call from the Chinese regulatory authority, and they said, would you please file your vaccine in China? And we pointed out that we were in the process of doing the required trials that China requires, specific China trials. We were in the process of getting the specific data, and we reminded them, please remember, you only allow one drug substance production site and one drug product production site. This is a 9-valent vaccine. We don't make it at just one site. We make different serotypes at different sites. And so a negotiation went on. The negotiation went on for a week. At the end of the week, the Chinese approved our vaccine with an American filing. We didn't even write a Chinese filing. Two work days after that, we got an order for 50 million doses. We didn't have 50 million doses to spare, and so now we are on a graduated path to provide, as part of that, 240 million, the doses required for China. But these are the sort of complexities that we face, and so now imagine we are, as I said, we're blowing out our supply chain. We're multiplying the lines we have by a factor of 3. We have to go and make sure that we do the appropriate technology transfer for those lines, and in order to not delay regulatory approvals of these new lines, not change a thing. If any of you are familiar with the whiskey business in Scotland, right, they have these big copper stills, and over time they get wears on the stills and they put a patch on the still. If they ever have to replace the still, they replace the still with the patch because they fear that if they don't, they'll get a different tasting whiskey. And while it sounds simplistic, we feel like we're in the whiskey business. So this ends our first tale, and we look forward to maybe coming back sometime in the future and talking about how successful we were at eliminating HPV-related cancers. There we go. Up. So let's go to the second part of our tale, which is about speed. Now, if you recall, we talked about general cancers. We talked about the fact that some cancers can camouflage themselves from the immune system. And about 8 years ago was the first real work done to say, well, can we somehow block these PD-1 receptors so that T cells don't deactivate, can see the tumors and attack those tumors, and let the immune system do its work against cancerous tumors. Like many other companies, we developed an antibody, it's called Keytruda, that does that work. We were very excited, and this is like 2011, very excited about the prospect for this drug. We took it into clinical trials. We did our phase 1 clinical trial, and not only we, but the FDA was absolutely amazed at the results from our clinical trial. And I'll pop back to this slide in a second. But I'm trying to give you both a pictorial, which is pretty powerful, but also the graph, right? So on the left-hand side, this is a patient with melanoma. That was the first cancer that we looked at, is to say, can this drug be effective in immuno-oncology? You can see already the tumorous lesion here. This patient was, at that time that the first picture was taken, was estimated to have about 3 to 4 months. You can see what the state of the patient's lesion was at 90 days. And we can project that over onto the Kaplan-Meier estimate here, which is a way of looking at progression, and this is, in this case, progression-free survival. How long do patients continue to live without having their cancers progress? The bottom line is best available therapy, which was platinum-containing chemotherapy. The top line is patients instead receiving this immuno-oncology agent, Keytruda. And this here is, as I said, the progression-free survival, the percent of people who have that survival. This is months. And as you can see, as the graph widens by 15 months, there's essentially nobody left in the control case with progression-free survival, and yet we're at 50% or more with the immuno-oncology agent. And this, in a word here as stated, is hope. This is hope for folks, and this was the data that we had from our phase 1 clinical trial, and that led to a race, right? Because when the FDA saw our phase 1 data, they said, don't wait for phase 3, let's get this thing filed. And so if we look at the statistics, we started in 2011. From the time that we started our trial to approval was 3 years. And we'll talk about what that means from a development perspective in a second. We see that from the end of phase 1 to ready to file was 1.5 years. FDA approval, and this is where scales can kind of trip you up, that's 8 months. So it was 8 months review time and approval based on phase 1 data. Alternatively, looking at it, in 2011, when we started on this journey, we had one clinical trial site, 20 patients. By 2014, given the results we had seen at the end of the phase 1 trial, we now had 24 clinical trials going in 30 different tumor types with 6,000 patients. So exciting was this drug. I'm going to go to a kind of a example timeline, and I want to say, when we're sitting on talking about the frontiers of biotechnology, let me be very clear. I am not in any way going to represent the Keytruda case as being on the frontiers of technology in biotechnology, right? And I will say that at the time we started developing Keytruda, Merck had never produced a commercial monoclonal antibody. We were a vaccine and small molecule company, right? And so we were fortunate. We'd hired a couple of people from biotechnology companies. We had a lot of people in vaccines who knew the tools, they knew the unit operations, but nobody who'd really ever, not as a whole, making monoclonal antibodies. And this was the group that was set forward to say, hey, we're going to do something that's pretty incredible. A typical timeline is shown here, where you can see that from a manufacturing perspective, as we start saying, well, where are we going to site this thing? How will we do tech transfer, characterization while we're doing phase 3? How are we going to then validate our batches and do our regulatory filing? You can see the timeline here, typical timeline, 6 years, 7 years, right? Which is not by any means lightning speed, but it allows you enough time to do these things in series, right? So here we had the Merck group who wasn't particularly experienced, and our timeline looked a lot different. Our timeline looked like this, right? So here's approval. So first patient was in 2011. We in 2012 and 13 needed to be doing technology transfer while doing process characterization. So we were transferring a process that was not fully characterized, doing performance qualification and regulatory filings in 2013, just 2 years after it first went into patients. So as you can imagine, this is not your high-tech, bleeding-edge process, right? I'll be the first to say the backbone of our process, it's a 1990s process, right? It's your basic 20,000 liter stainless steel cell culture, small grams per liter going into 3-column downstream processing. It's by no means technologically advanced. But there are times when, as biochemical engineers, it's not the technological advancement that's the challenge. And in this case, it was the speed. And we all had in mind that patient with the lesions, right? Because we showed that picture around to say, look, this is why we're going to be doing the work we're doing. And this is why we're going to go on this timeline. And you can imagine, particularly for people who had not a lot of experience in making commercial antibodies, the incredulity of the people doing this, right? Saying, how are we possibly going to meet these timelines? But let me show you how much more difficult it was than just shown here. Think about the technology transfers that were required. So we had a drug substance site that was a small pilot plant in Switzerland, right? It didn't have a lot of capacity, but it was what we had. And so the first order of business was, how do we convert this to a commercial facility so we at least make something for launch because patients need it? So that's where we first started. And there, that was not a technology transfer issue because after all, it's the site where we made the clinical material. This instead was how do you take a pilot plant and make it a commercial production facility, following all the rules that are required for a commercial production facility. But we knew that wasn't going to be enough. So we also began tech transfer to a first and then a second CMO. And our first CMO was an interesting choice because sometimes desperation is what it is. And so our first CMO was actually a competitor pharmaceutical company that had a lot of extra capacity because they had a facility that they weren't really using. And so we said, hey, what do you think? And of course, you know, now they had already caught on. They were developing their own anti-PD-1, but they said, it's an empty facility, let's go in and do it. And of course, we were inexperienced going in and do it. The one thing that we didn't know is they had less experience than we did. So it made it really challenging doing this technology transfer, but that's what we had to do. And we did this initial tech transfer. We did have one site that had one cell culture bioreactor, and we said, let's get them involved too. So we started doing a tech transfer there. And so we went along, and you know, this was the drug substance. One also has to ask, well, what form was this given to patients? When we first started this saga in phase 1, we did kind of the standard phase 1 approach. You don't put a lot of time in developing the formulation yet because it's phase 1. You don't know if it's going to last or not, right? So it was a lyophilized form, not the form we would want to go to in the marketplace. But since we filed on our phase 1 data, the only thing we had in the clinic was the lyophilized form. And so we had to do a tech transfer to a lyophilization site within Merck, while at the same time developing a commercial liquid image and getting ready to transfer that to another site so that we could get that approved as soon as, or file that as soon as the lyophilized image was approved. So you can imagine a lot of different technology transfers, and our supply chain complexity ended up in this very short period of having 4 active drug substance sites, 2 active drug product sites at launch, one of which was making the lyophilized, one of which was preparing to make the liquid. Now remember, it's not just about process. We also have to have the analytics to say we know what we're making and we're making it. And so from an analytics perspective, the specifications for the quality attributes, and we'll talk about this a little bit more in a minute, were based on very few batches. And those of you who've been in the business know when you're basing specifications on a really small n, there's one thing that spells, and that's trouble. We'll get to that in a minute. And the last piece was our analytical methods coming right out of phase 1. Robust would not be how you would describe them. And we'll talk a little bit about the ways in which methods cannot be robust. But the bottom line is we had a lot going on in a short period of time with an inexperienced group. And one thing that you can't forget when you're dealing with the biopharmaceutical industry is, in the end, you actually have to file this and get regulators to approve it. And if you've been in the business, you know regulators around the world, they all have their different time frames of approval. They all want something a little different. So you've got to cater to all of them. And once you put in the file, you may think, oh, we're done, we can rest. No. Once you put in the file, that's when the real fun begins, because that's when all the questions come back from regulators. And so you have to respond to questions, so the fun never stops. So real challenges. Now the question would be, why do all this? Why go through this insanity? If we had followed the standard pathway, and we've done some estimates, we would have ended up in 2018 having had our product approved. We estimate we would have about 6,000 patients treated. We'd be in about 12 clinical trials. We'd be approved for 1 cancer type. By going through this rapid approval process, by taking it to market quickly and continuing to do very significant clinical trials, we end up in 2018 with over 200,000 patients treated. Right? Over 12 million doses administered. We're now in over 900 clinical trials in 2018 with 14 approved indications. The amount of good public health we can create, the amount of sisters and brothers and mothers and fathers we can save from cancer is huge by putting on this kind of speed effort. We talked a little bit about supply chain, and I do want to address this a little bit because as engineers, you will often be called in to say, what do we need to make as our factory? How big does it need to be? What do we need in terms of equipment? What are we planning for? And while this was really speedy, this was really challenging. At the time we started our technology transfer, you've seen the compressed timeline, but we faced a whole boatload of uncertainty. The dose had a range of 7x at the time we started this. And we didn't know which was the right dose. Was it the high dose or would the low dose be sufficient? But you got to plan for both, right? In addition, at the time we came out, BMS was ahead of us with Opdivo. We didn't know what would the competition in the market look like. And of course, everybody and their brother and sister were also developing an anti-PD-1. How lasting would this product be? We also didn't know how many indications it would be good for. We filed for melanoma. The second thing we went for was lung, but we didn't know would it be good beyond melanoma. We had no data to say that it would. We had no data to say that it wouldn't. What do we plan for? And finally, how many markets will we go into? How many markets will be receptive? How many markets do we want to file in? How much will we have supply for? All of these uncertainties play into, well, what kind of supply chain do we want? And we decided, given the potential value this has in patients, we have to go big or go home. Right? So let's make as big a supply chain as we think we might need should upside occur on all of these uncertainties. And so we had this accelerated development program with this uncertainty on dose and growth. Again, we had limited biologics drug substance capacity within Merck. And we did know, though, that whatever we would do for key nodes, we wanted to be dual sourced. Because if one source had a problem, a mycoplasma infection, whatever, we could not go without. And so in creating this supply chain in 2014, and you'll see the little Keytruda marker as it goes through time here, right? This was the supply chain we had. Even our cell bank, which is our IP, right? Our cell bank was external because we had no cell bank for cell culture for proteins. We had plenty of virus cell banks, by the way. You want to come and get some virus, sure, but nobody wants virus in their protein. I don't know. From a drug substance perspective, we started off, remember, with that pilot plant in Switzerland, and we went to this external competitor to have them help us out. This was our drug substance. Drug product, the lyophilized form, in one facility. We had one packaging facility, one. And of course, you know, might as well, if you're going to go in for a penny, in for a pound. A contract lab was doing the analytical work, the release work for certain assays because we didn't have labs to do it. Right? So this, in the business world, is called world of hurt. Right? And as you can imagine, the first presentation to the board of directors at Merck on, here's what we're doing for manufacturing for Keytruda, this presentation did not go well. Okay? But we rapidly evolved the supply chain. We did quickly bring online a liquid drug product facility. That was in 2015, more packaging. But that obviously was not enough. And it was interesting. We were having this CMO make all it could because we didn't know were we going to be at the low dose or the high dose. And around this time frame, the 2015, 2016, it was decided we would be on the low dose for all indications. So we actually had to tell them to slow down because we ended up with freezers full of drug substance. And we thought, well, we're sitting pretty now. We got freezers full of drug substance. But then 2016 came around, new indications were approved, and that stuff flew out of the freezer. We shut down the commercial work at this pilot plant and went with a very large and well-known CMO to bring on new capacity, which would give us adequate capacity, again, to meet what we thought were these uncertainties. We were still making the lyophilized form, but now we had filed the liquid form, more packaging, but still contract labs. And by 2020, by the end of next year, while we still have a variety of CMOs online, we will have a large facility in Dublin, Ireland that will be making the majority of our drug substance. We will have eliminated the last of the lyophilized product in the marketplace and now be fully liquid and having 2 locations, multiple packaging locations, and labs now fully within the Merck network. And then by 2022, we'll have our own internal cell bank, all internal, and you'll notice the amount of external has decreased significantly. This picture wins with the board. I also talked about analytical pieces, and I think it's important to talk about the analytics, right? And the first piece is specification setting, and I'm talking about specification setting when you have really small n, like you've done 5 pilot plant batches, right? But you've got to set your specs, and it feels over time a lot like this cartoon. So initially, agencies are willing to give you some leeway. We know you're accelerated, you only got 5 batches. This bigger range seems reasonable until we really fully understand what your process is capable of. And so that's where it was set. Specifications get tighter as you begin to prove the capability of your process. In our experience, they became incredibly tight. And I'll talk about why they became incredibly tight. If you look at this timeline, right, so initially we had this first approval, we had these wide specifications. As we made those first batches in Switzerland and at this CMO, you can imagine we could not afford to lose a batch. So we had engineers swarming all over these batches, analysts swarming all over the contract lab that's doing the release because we couldn't miss a step. And we got really good process response by having that sort of piece. And by the way, we didn't want to take any risks when it came to raw material variability. So we're using one lot, right? One lot of media feed, one lot of everything. Well, you can't do that forever. And in 2016, boom, we're now starting to see the normal variation in raw materials. We now don't have 1,000 engineers watching every batch, so you're starting to see normal variation in operation and normal variation in the contract lab. And we have a batch that goes out of spec. Ironically, it's within the first specs we had, but not in the specs that we were forced to take as time went on. We did get those specs widened when we finally had a refined specification set based on data, but really challenging, and like the line does, at times makes you want to jump out of the window. The other piece that was really important, and something that, again, our lack of experience, we didn't pick it up. Many of the assays we use for showing the potency of this antibody are specific to this antibody. And you're like, yeah, okay, I get that. But that means the reagents are specific to this antibody. And in research, they were like, okay, yeah, we need some specific antibody reagents. Let's go ask them to make some for us. Right? Well, the them was a reagent house which was not normally in the business of making reagents for commercial testing. Right? They're like a specialty house that makes reagents. And so, hey, we got some in. That's great. Those now become the source of these specialized reagents for the methods. And again, that spells trouble. Look at the graphs at the bottom here. These are 2 graphs from ELISA plots. What's interesting, you can see the difference between these two. Same method, same lab, same analyst, different lots of reagent. Right? And we had to go back and make a lot of these reagents ourselves, go back and completely retool the reagents for these assays so that we eliminated this type of variability. Rookie mistake, yeah, perhaps, but given the speed, something that was necessary. So as we look at where we are now, this is the 5th anniversary of the approval of Keytruda. We now think we have a much better understanding of its long-term variability. We're seeking to make clinically relevant specifications, and we feel like we have a well-characterized molecule and process. We've digitized our manufacturing, collecting data from all of the manufacturing sites and being able to visualize that data kind of in a minority report type style, so we can look at the differences between the facilities. If we put in some advanced process controls and are platforming our processes, however, from the time of that very first approval to correct all of the things we didn't want, will take us about 10 years to get to the right image, sustainable control, a higher yield manufacturing process in our own facilities. A real challenge, but necessary in order to get the product to the patients who needed it most in the time with which they needed it. So our work isn't finished on this one. We're still investing in our patients. One of the things that when you have a product like Keytruda and you're looking for more indications, it presents a real challenge in R&D. Because if you have $200 million, do you want to invest that in the next HIV product that you don't know whether it will succeed or fail? Or do you want to put that in a phase 3 Keytruda trial? The phase 3 Keytruda trial is the best bet, but then you end up being fully concentrated on Keytruda and not on the other products in your portfolio. Very challenging, but we continue to invest in new clinical trials and indications, building a wall of data showing the superiority of this product. We're going to new countries, creating new manufacturing sites, and creating a robust infrastructure. And I wanted to put kind of as a coda where we are now in 2019. Going from one trial in 2011, this year we reached we're actively in over 1,000 clinical trials. Those clinical trials are by and large combination trials where many of those trials are not Merck trials. There are other companies' trials who give Keytruda in combination with the product they're developing. We're now in 87 markets. We're approved in over 21 different indications. So we continue to find new and different tumor types that we are applying Keytruda to. And we've now treated over 250,000 patients. And just some pictures of patients. This patient actually works in the building next to mine. Have met him several times and shared the podium with him several times. And every time he gets a clean scan, I get a little email, scan is clean. When he first took Keytruda, it was estimated he had 2 months. And that was 4 years ago. So really great. But when I say we're not ending, some tales don't end. They have new chapters, and Keytruda will have a new chapter. We are working on a fully continuous process, our next generation process for Keytruda, featuring high titer perfusion-based cell culture, a full continuous downstream, and the necessary feedback control loops for that. This is a process that we've now run 3 times from the 400-liter scale in the cell culture. We've run 3 times for 25 days each through the continuous process. So we are getting ready to make our first GMP material with this continuous process. So that's a part of our investment in the future of Keytruda. I do want to thank folks who contributed to this, a variety of folks who contributed both to the presentation and hundreds of biologists, bioanalysts, biological folks, and biochemical engineers. And I'm hoping that our time on the frontier has been helpful. I realize I stand between you and costumes and beer. But I do want to say that out in the frontier, I think there are many tales. We've tried to do 2 parts of this tale today, right? One of complexity and one of speed. And I have to say that whether you're doing the speed or complexity, you don't start in a great place, right? When you start, oh, there we go. When you start, you feel that you're in darkness because you don't have a lot of knowledge. And how are you going to make this thing work? That darkness turns into despair, right? When you've worked for a while and it's still not working and all you hear is, where is my drug, right? And then you say, well, we're never going to be, this is foolish. We're doing this foolish errand, something so complex or so fast. How could we do this? And it leads to incredulity. I don't see how we're going to do this. We have nothing before us, right? It is the worst of times, right? But then you get some data. And the data starts to work, and then there's light. You start to see what you can see. That light turns into hope. Maybe we can actually pull this off. The hope turns into wisdom. And that wisdom comes not only from the hope of maybe we can turn this off, pull this off because we have that light, but we start seeing the results from clinical trials, and we see what these can do for patients. And with that wisdom comes belief. We can and will do this. And with belief, we have everything before us. It is the best of times. Thank you.
Excellent. We have time for questions, starting with Clark and then
Clark.
Martin.
Mike, I'd like to congratulate you on a truly magnificent presentation. And I personally find this very inspiring because we live in a time when parts of our society are totally dysfunctional. And we as educators should take at least some pride in hearing this talk because we have put together a system. And every once in a while we see the results that what we tried to do did impart some tools. Come back and tell us this magnificent story that's really very inspiring. And I just feel very good that you were part of the story here at MIT and come back and tell us this incredible success in helping people. So thank you very much.
It's the work of hundreds and hundreds of people, Clark. Right? But I appreciate the self-congratulatory nature of your comment.
You haven't changed.
We'll try to move the mics around for questions. Nubar.
Mike, in this series of speakers, you're one of the few, maybe the only, who has actually graduated from this department. And so it would be interesting to think about what one kind of, not lesson, but what area that you learned here that contributed to what you're saying to us today, and what one thing you wish you had learned here, what one course, one area that would have served you better to be even better than what Clark described you to have been.
So what made me better, right? The department, and still has, I believe, a great Europe program, yeah? Okay. Take advantage of the Europe program. I have to admit, I took over advantage of the Europe program at one point, having 9 Europes. I learned a lot about management skills from having those Europes, right? I learned what it takes to give people enough so that they can go and be self-directed and do what they need to do without being micromanaging. As I had one Europe famously tell me, "Get out of my face, Thien. You're in my face." Right? That was a really valuable management lesson. Nubar, if I ever say the one thing that I wish I had, boy, I wish I had learned how to do cell culture. You know, as Alan will tell you, I was a downstream guy, way downstream. And so while I could look at my friends who all did cell culture and they were having great times being here all times of the day and night, weekends, you know, praying that they didn't get contaminations, I couldn't relate to it. And I have to admit, I think I would have been better served had I done some cell culture. You know, they say I don't have any culture.
So thank you for a very nice talk. So I just wanted to ask about the role of kind of fundamental understanding in this kind of work, because when you go faster, you have less time to kind of figure out what's going on. And I was particularly interested on the first part of your talk on just this sort of packing problem of these, I guess, capsomeres. I've noticed there's occasionally 5-membered rings. These are actually pentagonal things. Usually soccer balls have 6. Mm-hmm. How well did you understand all that stuff? You know, and was it necessary? You know, because there's sort of a trade-off between actually understanding, because you realized you had to break them apart. So take away electrostatics, you put it back. Electrostatics really want 6; it doesn't want 5. And so there's I think some interesting fundamental questions there, which you can address by trial and error, and or by trying to understand it.
Between the first and second rounds of vaccine production or vaccine design, there was work in the literature that helped illuminate some of the fundamentals of this. But I have to say, at least our first go-round, it was about using the fundamentals like blunt tools, right? Yeah. We know we need
and there's nothing wrong with that.
Well,
I'm just asking what the role is in this.
It's not that there's nothing wrong with it. It's what we had to do, right? And at the time, we didn't really have the time to be able to do the fundamental work necessary. And I'm going to argue, I don't think we had the tools, right? And some of the tools we had to develop while doing the development work here. But I think in order to make something that we could prove to ourselves we could do reliably and had the right immune response, we didn't need the fundamentals, right? Clearly, over time, others have been looking at the fundamentals. We have a whole program looking at the fundamentals so we can truly understand the mechanisms involved here. But there are times when you don't need that to be able to be successful.
Thank you.
Oh, don't let her ask a question.
Thanks for the great talk. I have 2 questions on your multivalent vaccine and the cell culture for it.
Yeast.
So, yes, I know it's yeast.
Okay.
It's Saccharomyces, right?
Yes.
Okay. So my first question is, were the cell lines locked before you tried to develop your reactor processes?
Yes.
Okay. So do you think that if you had considered how the cell lines would perform in the reactor process, that you might be able to get more consistent processes across the serotypes?
I think that would be the case, but we didn't have line of sight to that. And we had to move faster than would allow going back and re-looking at the cell lines.
My other question is, did you ever try to make multiple serotypes in a single cell line?
Not that I know of, but I have to say I can't say that for sure.
Okay, thanks.
Thank you.
Yes.
I can't help but ask a really picky question.
Oh, please.
One of your early HPV slides showed that pressure has a very substantial effect, or can have a substantially...
This is pressure on the reactor, right? Back pressure on the ferment.
Exactly. And that astonished me. I didn't think pressure would be important at all. Why is it?
We didn't either, And that's one of the areas where we don't have the fundamental understanding, but we certainly know from a experiential basis that it has an impact.
Thank you.
Mike, it was terrific how you brought the uncertainty and the mistakes and the errors into the talk, because usually we don't admit to those things, and everything looks like a nice rosy picture. You had a
You know me better, Charlie.
I've seen this before a few years ago.
Yeah.
You had a real challenge to deal with the multiplicity of regulatory authorities, not just the FDA, but
Still
beyond.
have.
And how have you used your scientific understanding of the process in order to facilitate that conversation and perhaps accelerate that conversation with the regulators? So I'm looking at that collaboration.
So I would say actually it's kind of a spectrum, Charlie, that initial interactions with regulators, because we were on such a speedy path, they would allow well-put scientific argument and theory to help, being augmented by data. As we've gone on in time, don't bother giving us theory anymore, give us the data. And so as you can imagine, we have an ongoing development work that goes on not only on the first generation process, but an improved process that we'll be putting in place in the next year and our next generation process, continually adding to the data. And one of the things that we have now that's really powerful is we have data from 4 different commercial scale sites on the process and the methods that provide a huge amount of data for us to be able to say to regulators, look, in adding this new site, it is completely comparable. Look at every aspect of the data to the 4 sites we've already put it into. And that's really had to be our strategy. I will say, though, that we do map out the regulatory strategy some years in the future. And we have, when mapping that out, it isn't as much of what scientific data do we need as what is the turnaround time for a regulator. Because as you can imagine, as we put changes in, the US may approve it like that, right? But Japan may take 1.5 years to approve it. You fragment your supply chain, and it gets to the point where you may fragment your supply chain. You're no longer running the process that a country had approved and hasn't approved anything else, and now you don't have anything to supply. So that planning, all of those pieces have to go together. That part of the planning where you don't want to over-fragment your supply chain can actually limit the speed at which you can put in technical improvements, because that will over-fragment the supply chain. Tough stuff.
All right, let's take one question in the back. Malin's got it.
Paula told me, by the way, if we're here long enough, beer will start coming down from upstairs.
That sounds fantastic. I guess the flip side of the coin to expanding manufacture and scaling up manufacture is dose sparing. So I was just wondering where you see alternative adjuvants and different delivery routes for HPV vaccination going forward, such as mucosal immunization, transdermal delivery?
So from an adjuvant perspective, before we go to different types of delivery, we have been examining actively different adjuvants other than the monoaluminum phosphate. So far, we haven't come up with anything better, but we continue to remain open to that. And there are a lot of adjuvants that are available, I'm going to say theoretically. They either have worked in other vaccines or they've been developed and they should have all the right properties. And we've taken numerous of those and put them into animal studies and some that made it all the way to people and failed in clinical studies. When it comes to different delivery mechanisms, we're also looking at that particularly, believe it or not, for low-cost countries, right? And we, in our examinations of those so far, have not come up with a delivery mechanism that matches the needs from a stability perspective for the virus-like particles. But we continue to be on the lookout, because if we do want to eliminate HPV-related cancers worldwide, we have to be able to get it to places and deliver it in ways that aren't the traditional syringe or vial into syringe.
So does that include like solid formulations for improved stability? Have you seen benefit there?
We have not seen any benefit from solid-based formulations. I have to say that we have looked at some. I know of 2 that we've looked at, and we didn't get the results that we needed to see. I think we continue to be open, and that's one of the the things with vaccines that is a challenge. The more people your vaccine goes into, the higher the bar of proof that the vaccine will be delivered in the way that it was, the higher that bar is. So you can imagine for measles, mumps, and rubella, right, we're using a process that is 45 years old, and the FDA has told us, look, it's been in billions of kids, you don't dare change it. So challenges there as well. Great questions, thank you.
We would have time for one more student or postdoc question, if we have one.
Oh, I love that, student or postdoc question, okay. No? Favorite color is blue.
We have one over here. Let's see.
Thank you for the great talk. Actually, I'm not really coming from the biotechnology background. I'm really coming from chemistry. So I got very interested about your second half of your talk when you're talking about the speed of the immunotherapy. So what do you think is the rate limiting step for this kind of a speeding process?
Oh, that's a really great question. I think the rate limiting step is the generation of sufficient clinical data. Right? In the end, it's always, if you are a developer in the biopharmaceutical industry, let's be really clear. Your job is to stay off the critical path. Clinical should always define the critical path, and as it was here. And our job was to get off the critical path once the FDA said that data is great, file.
I see. So, but in your talk, you are sort of saying like you are speeding up the sort of having a new strategy compared to the typical.
So that we remained off the critical path for filing approval and getting doses to patients.
I see. Okay. Thank you very much.
Before we say goodbye, I'd like to present.
By the way, that's Dickens, in case you'll guess. Right?
Aha!
Okay.
A little bit of packaging here. We'd like to present to Professor Michael Tien.
Professor?
There's professor on there.
Oh, that was
You've been
great.
elevated.
Thank you. I love it.
We would like to present to you the DIC Wang Frontiers in Biotechnology Lecture Award. Thank you so much, Mike.
Thank you, Paula. Thank you so much. Thank you.